Okay, we're going to take a little time out here since we are discussing the small intestine and things that go on in that first section, the duodenum especially. We're going to take some time to discuss two accessory organs of the digestive system, the liver and the pancreas, which provide substances that are critical for the digestive processes that happen in the small intestine. So we're going to head over to the liver and also the gallbladder for a few moments. I keep leaving out the gallbladder. The gallbladder is important too, so we'll be discussing that in this video lecture as well. Both of these are considered to be accessory organs of the digestive system, so they're not part of the alimentary tract, but they supply substances that facilitate digestion, help it out. Now, the liver has lots of non-digestive functions as well, so don't think that that's the liver's only role. The liver is an extremely important organ for the body. The liver is over on the anatomy side. We'll talk about how the liver is divided up into four lobes. The right and the left lobe are the big ones, and then you've got two smaller ones called the caudate, which kind of looks like a tail, and then the quadrate, which looks kind of like a square or a rectangle. Many functions, as I mentioned. The digestive function that it has is producing bile, and the role of bile is to emulsify fats that are coming in from the stomach into the small intestine and need to be digested. And we'll talk about what that means shortly, but if you think about it, you guys know fats and water don't mix, hydrophobic, hydrophilic. So fats that we consume in our diet tend to be present when they arrive in the small intestine. They're like in little fat globules. Well, it's very difficult for the enzymes that need to digest those fats, down at a molecule type level. It's hard for those enzymes to access those those fat molecules if they're all hanging out together in a big glob. So we'll see that bile kind of functions almost like your dishwashing detergent of your digestive system and helps dissolve those fats and make them easier to break down. The gallbladder, its main function is storage of bile. So the liver, as we'll see, actually makes bile. Excess bile is stored in the gallbladder, and the gallbladder is like this little thin muscular sac that contracts and squeezes bile down into the small intestine when it's needed. All right, so this is being covered over on the anatomy side as well, but I'll just do a little quick review here. Since we're talking about the digestive functions of the liver, here's the left lobe of the liver, here's the right lobe. The other two are on the underside, back side of the liver. Here's your gallbladder over here. And as we'll see, the liver is divided up into many liver cells. Those are called hepatocytes. And they have many functions. One of their functions is to produce this substance called bile. And so it winds up draining from all these various liver cells. And there are lots of little tiny channels that really only contain bile. And those lead to hepatic ducts from the right side and the left side that have collected that bile. Those come together into a short duct called the common hepatic duct. Okay, so the right and the left come together and form a common one. Excess bile, or bile that needs to be stored until it's needed, moves up this way, up the cystic duct, over to our gallbladder. It gets stored in here. This is a little thin muscular sac, and when bile is needed down in here, here's the duodenum, the first part of the small intestine. The stomach would be sitting right in here. It's been removed, so we can see these structures underneath. This will contract, the smooth muscle in there will contract and squeeze bile this way down the cystic duct, and then it travels down this tube. Now, this is called the bile duct or the common bile duct. So there's lots of bile ducts. You just have to practice with it and learn the different names of them. And then that bile duct or common bile duct passes behind posterior to the pancreas down into the pancreas. And right here it is merging with a large duct in the pancreas that contains digestive fluids that the pancreas is making. So those two ducts come together. They form a compartment here called the hepatopancreatic ampulla, An ampulla is a little fluid-filled compartment. The name sounds like ampule, if you've ever heard that. A lab ampule is a little glass vial that stores fluids. So you actually have a combination of bile and pancreatic fluids that are stored in that little widened area. And then there is a sphincter right in here in this area called the hepatopancreatic sphincter, which is a circular, smooth muscle type structure that will relax and open when bile and the pancreatic fluids need to be released in here into the duodenum where your foods are being digested. So that's sort of the architecture of those different structures. Again, here are the names of the bile ducts, as I just described on the last slide. Okay, now what's going on in the liver microscopically? The liver is obviously a very important organ with lots of functions. Very large organ, and it's divided up into areas called liver lobules, and they look like hexagons, 3D hexagons, and these are considered to be the structural and the functional units of the liver. And again, the cells in there are called hepatocytes, so the liver is jam-packed full of these types of cells, and they have many functions. In addition to producing bile, they handle. Now remember, when we were going over the cardiovascular system, we talked about how blood that is draining off of the stomach, the small intestine, the large intestine, and the spleen all travels into the hepatic portal vein. That's deoxygenated blood, but it's rich in nutrients, and that blood goes to the liver through that hepatic portal system, and it gets distributed to the hepatocytes, and the hepatocytes take up and process these nutrients, vitamins, various things that are coming in from the digestive tract. The liver stores some of these things. The liver uses them as building blocks to create various important things that the body needs, such as your clotting factors. The liver produces a lot of proteins that are critical for your bodily functions, especially in the blood, like your clotting factors. Also some proteins that are important for immune system function as well. And as we'll see, each of these liver lobules has a central vein that's running longitudinally up and down like this, that collects the processed blood. Remember how we were talking about the blood being filtered in the liver? The filtered blood, after the hepatocytes have had their way with the substances in there, drain into a central vein in each of these lobules. So this is actually a diagram from your textbook, which is showing you if you slice the liver open and you were looking at a two-dimensional slice of liver tissue under the microscope, you really would see that you can really see these hexagon-shaped lobules in there. Almost looks like some sort of kitchen tile. So the darker areas are where your hepatocytes are located, and those are full of blood. The liver is, very rich in blood. The lighter shaded areas in there, which almost look like the grout between the tiles, those are connective tissues which divide each of the lobules. And you're going to have blood vessels, lymphatic vessels, and so forth, the usual types of things that you find in connective tissue fillers passing through there. And then the white spot in the middle over here, one lobule is blown up. That's where you're going to have your central vein. So blood is basically coming in this way, passing through these hepatocytes, or getting filtered by the hepatocytes that are located here in the lobule, and then the filtered blood drains into these central veins. The central veins are going to take that blood and deliver it to the hepatic veins. If you guys remember, the hepatic veins then connect into the inferior vena cava. So finally that filtered, deoxygenated blood makes its way back to the heart. Now here's a really nice diagram from your textbook, which is showing you what one of these liver lobules looks like. It's one of these hexagon-shaped structures, and as you can see, they are actually 3D structures. They're not flat little tiles for real. They actually are a three-dimensional structure, And so what they're showing you on here, here's a portal vein. So the portal, now you have the big hepatic portal vein that's bringing the deoxygenated blood in from the spleen and the digestive organs. But then that's going to branch out into smaller portal veins that are going to take that blood throughout the liver. And then there are going to be exits off of these portal veins, like you see here, which are going to take this blood up into different parts of one of these liver lobules. Okay, and then scattered around all these blue vessels that you see here, those are liver sinusoids. If you guys remember from when we talked about the different kinds of capillaries during Unit 3, sinusoids are very leaky capillaries. So the sinusoids are taking that filtered, the blood coming in from the portal veins, and it travels into these sinusoids that are scattered all throughout the liver lobules. And those capillaries are very leaky, which is good because the substances that are in there need to easily get out so the liver can process them. All these hepatocytes you see there can do their jobs. And the hepatocytes are also making substances. So another good thing about having these leaky sinusoid type capillaries in there is that they can easily pick up substances that the hepatocytes are making, such as your clotting factors, such as other blood proteins that play important roles in immunity. The hepatocytes also create little particles that transport fats and cholesterol throughout the body. We'll talk a little bit more about that at the end of the unit. So the key point is there are lots of substances that need to move in and out of the blood around these hepatocytes so the hepatocytes can process them. All right, so after the hepatocytes have dealt with these substances and sent whatever they need to into the blood that needs to leave the liver lobule here, the filtered blood winds up here in the central vein. So the blood is essentially moving like this across these liver lobules through those sinusoids and towards the central vein. And then the central veins will lead to other veins that connect to other veins. We're not going to worry about the names of all these different liver veins, but those are, the key point here is those are eventually going to lead to your larger hepatic veins, and you have those coming in from the left and from the right, and those connect to the inferior vena cava. So this is where our liver filtration story that we talked about during cardiovascular has taken place. All right, then another thing I want to point out here on this diagram of a liver lobule, you guys can see the little green spots scattered around on the diagram. Those are representing sliced open bile caniculi or canaliculi. You've heard that term before. You heard it when you studied bone anatomy in biology 201. The osteocytes in your osseous tissues have little projections on them that travel through canaliculi, little tiny canals inside your bony tissue that allows the osteocytes in your bones to contact each other and exchange nutrients and gases with each other. So here you're seeing canaliculi. Again, it means a tiny microscopic canal, and those are collecting the bile that is being produced by these hepatocytes. Again, that's one of their functions, and those lead to larger bile ducts that you see over here. And so you have these scattered all throughout the liver. They're eventually going to converge into the left and the right hepatic ducts that contain bile, which then lead to the larger duct. Let's go back here and review on our picture. Those eventually lead to the common hepatic duct that you see right here, and then that excess bile travels up the cystic duct to the gallbladder for storage. And again, keep in mind, you've got these liver lobules scattered all over the liver. You're looking at something that's down at a very detailed level. A little bit more about the hepatocytes themselves. These are very metabolically active cells because they have so many different jobs. If you looked inside them, there would be large numbers of internal organelles. Organelles, if you guys remember from earlier biology classes, those are membrane-covered compartments that have specialized jobs, such as digesting substances, detoxifying substances, modifying proteins, transporting and secreting proteins around the cell or up to the cell surface, and you have lots of those different kinds of things going on in your hepatocytes. So the hepatocytes, as we've already mentioned, they're going to those cells process blood-borne nutrients. So if you guys remember, even back when we were studying endocrine, the liver stores glucose molecules as glycogen. So it links those together in chains, glucose, 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 etc. That creates glycogen, a type of starch that is stored in your hepatocytes, so that when you need to break it apart and release these individual glucoses back into your bloodstream, you have a source of glucose to do that. And that's just one example of things that the hepatocytes do. They store fat-soluble vitamins. We're not really going to have the opportunity to discuss vitamins in any great detail. You guys know that you need lots of different vitamins in your diet, and most of them are assist enzymes, and that's why you need them. They're called coenzymes because they help your enzymes carry out important chemical reactions. And our cells don't make vitamins. We have to take them in from our diet, and so many of them have to be stored. Fat-soluble vitamins are ones that don't dissolve well in water. They're hydrophobic, so they need to be surrounded by oily type substances. So those are stored in the hepatocytes as well. Your hepatocytes detoxify various things, including alcohol. Not that any of you would drink alcohol, but if you did, the liver takes in that ethanol alcohol, and there are enzymes in the liver cells that break it apart and convert it to non-toxic substances. Assuming you don't overwhelm your liver with excess alcohol, and then that becomes damaging to these hepatocytes, because alcohol is toxic for them just as it is for other cells of the body. So if you drink too much alcohol, the hepatocytes, which are programmed to take a lot of this alcohol in anyway, can wind up losing their functions or even dying because of that alcohol exposure. The hepatocytes make a lot of bile every day, 900 milliliters. So that's 9/10 of 1 liter. And you guys know how big a 2-liter bottle of Coke is. So think almost half of one of those. The liver is making that amount of bile every day, which shows how important it is. Some homeostatic imbalances of the liver. Hepatitis, and all that means is inflammation of the liver. Anytime you see itis on the end of something, that's inflammation. And remember, from when we discussed the innate immune system, what triggers inflammation? Infection or any kind of tissue damage. Injury, burning, sick cells, sick as in injured, unhealthy cells, those types of things will trigger inflammation and all of the various symptoms associated with that. Now, when people say they have hepatitis, they're usually referring to some sort of a viral infection like hepatitis B virus, and there are other hepatitis viruses as well. Those viruses infect the cells of the liver, and they wind up being damaged or even dying because of it. Drug toxicity. The liver detoxifies lots of things that we get exposed to, even things like pesticides and other kinds of chemicals out in the environment, drugs that we take. They don't last in your body. And I'm not necessarily referring to recreational drugs, any type of drug that you take. The liver is usually the organ that has to take that drug and break it apart, and remove it from the bloodstream so that it's not there permanently. So the liver is susceptible to damage from various kinds of drugs. And not that any of you are planning on going out and eating wild mushrooms, but poisonous wild mushrooms, the reason they're poisonous is because the toxins accumulate in the liver and damage the liver cells. There is a condition called cirrhosis of the liver that you've probably heard of before. And that's where the liver becomes fatty and fibrous, and it's due to long-term chronic inflammation. And this can be caused by hepatitis, like where you've had an ongoing, some of these hepatitis viral infections last for a very long time. Your immune system is not so good at eliminating those viruses. But usually when we hear about cirrhosis, it's due to alcoholism. So the constant exposure of the hepatocytes to alcohol kills or weakens many of them. That triggers long-lasting inflammation inside the liver, and so a lot of the liver tissue gets converted to fatty, fibrous type scar tissue, and that can put pressure on blood vessels in the liver and squeeze them, and that can actually lead to portal hypertension. We have high blood pressure in that, and high fluid pressures in the liver. You can get a liver transplant if your liver winds up being severely damaged for some reason, but livers for transplant are pretty hard to come by. So let's talk a little bit more about bile. So bile production is the one truly digestive function of the liver. And one thing I want to point out to you, since we're going to be talking about digestive enzymes shortly, bile is not an enzyme. Enzymes are machines, basically. They're proteins, but they function kind of like machines that do chemical reactions. They make it more likely that particular chemical reactions are going to take place. So like when we talk about glucoses being linked together to make glycogen in the liver, you need enzymes to make that happen. There are enzymes that take glucose molecules and force them to bond together in chains so that you have glycogen. Or when we're talking about proteases that break down the proteins in the foods that you're consuming, those are enzymes that take the proteins and break the bonds between the amino acids. They force those types of chemical reactions to take place. So that's what enzymes do. Bile is not doing that. It's not breaking down or building anything, but it helps the enzymes in our digestive tract that are going to break down those fats. And it does that through a process called emulsification. There's the word right there. Or we can say that bile emulsifies fats. All right, and this is a pretty good little picture that I found that shows this. So bile contains, the molecules in bile that really do this fat emulsification are called bile salts. And bile salts are cholesterol derivatives. And cholesterol is a type of lipid. Okay, so those are hydrophobic. But these bile salts are modified so that they have a hydrophilic portion and a hydrophobic portion. And so in green here, these are supposed to be our bile salts. And so over here on this side of the molecule, that is hydrophobic. Well, my pen's not going to write that far. Hydrophobic over here on this side. And then this face over here is hydrophilic. And so because of that, what happens is the hydrophobic portions of the bile molecules, you can get a bunch of them together, and they wind up surrounding fats. And the diagram there says triacylglycerol. which is more or less another term for triglyceride. Most of the fats that we consume in our diets are triglycerides. They're built from glycerol and 3 fatty acid chains. So those are hydrophobic molecules. And so these bile salts, what they do is they surround these lipid molecules. Because their hydrophobic portions love to interact with those fatty molecules, and then the hydrophilic portions over here on this side love to interact with the watery fluids that you have in your digestive tract or throughout most of the body. So by performing this function, what they're doing is they're making these fats more accessible to lipases. So the little red heart-shaped molecule things that you see there are supposed to be lipase enzymes that are going to digest the fats. So if you think about it, if you have some big glob of fats coming in to your digestive tract out of the stomach and into the small intestine for digestion, the bile salts, what they're going to do is help break apart those fat globules into smaller fat globules, and they do that through this process of emulsification. So in green I'm drawing in my bile salts there. The bile salts are able to surround smaller, much smaller droplets of those fats to assist with their being digested by your lipase enzymes. And that's what we call emulsification. So instead of having a big glob of fat sitting in a watery-based fluid, it gets broken apart into much smaller little parcels of fat or droplets of fat. And those can now be accessed by the lipase enzymes that are going to digest them. So that's called emulsification, or in a sense you're helping to dissolve these fats in water. And it is very much like what we just talked about here with the bile salts. That's very much like what the detergent molecules in your dishwashing liquid or other types of soaps do to fats. That's why if you drop some dishwashing liquid in a pan full of water and grease, the grease tends to disperse because it's being emulsified in a very similar way by those detergent molecules. Some other substances in bile. Where does it get its color? It has kind of a yellowish green color, and it is an alkaline type solution as well. Bilirubin, if you guys remember that from studying the blood back at the beginning of Unit 2. Heme, which is part of the hemoglobin molecules in our red blood cells, as those old red blood cells get broken apart and digested by your macrophages, the hemes get converted to bilirubin, and the bilirubin is excreted into bile in the liver, and then the bile gets released into the digestive tract, and bacteria in your digestive tract. You have tons of bacteria in your intestinal tract. They take the bilirubin and convert it to stercobilin, which has a brownish color and actually gives your feces the brown color that they have. I'm sure you always wanted to know where that brown color came from. Some other things that you'll find in bile: cholesterol. And bile salts are actually made from cholesterol, so it's not a surprise that we have cholesterol there as well. You do have some triglycerides and phospholipids. Those are types of various types of lipids, and some electrolytes as well are all mixed together to make the bile fluid. All right, then a little bit about the gallbladder. Again, that is a thin-walled muscular sac, and it stores bile. It actually helps concentrate it. It removes some of the water that's present in the bile when it comes out of the liver. And when needed, in the gallbladder, the smooth muscle tissue in the wall contracts, squeezes the bile down the cystic duct, which will then take it into the bile duct. And the bile duct is going to lead down to the pancreas and that hepatopancreatic ampulla we were taking a look at, where the bile will sit waiting for the hepatopancreatic sphincter to open up so that it can enter the duodenum and start dissolving the fats that are coming into the duodenum from the stomach. Okay, you can, if you hear about somebody having gallstones, another name for those is biliary calculi. Calculi is a term for stones as well. These can develop if a person has high cholesterol levels and too few bile salts. Because bile salts will help dissolve and emulsify cholesterol as well. So if you don't have enough of them and you have a lot of cholesterol in your body fluids, these cholesterol molecules can actually coalesce and form stones, these gallstones. And they can actually wind up obstructing the flow, blocking the flow of bile out of the gallbladder. If the bile is taking away the bile is taking away the bilirubin, okay, that's coming from your hemoglobin, your heme portion of hemoglobin that's being recycled from your red blood cells. If you block the exit of that out of the liver and the gallbladder, all this bilirubin winds up building up in your bloodstream and body fluids, and that's when you can become jaundiced, where people have that yellowish color in their skin and other tissues. That's due to the buildup of those substances because they're not able to exit the liver properly. Gallstones can also, they literally do form crystals, and those can be sharp so they can poke against things, and that can wind up being painful. There are treatments for these. Some of these are drugs that can help break the gallstones down. In other cases, they'll actually use ultrasound, sound waves. That's called lithotripsy, to try to zap them and break them apart. Lasers can be used as well, and in more severe cases, you might have to have a more invasive type of surgery to treat those gallstones. All right, in lecture number 6, we're actually going to continue our detour away from the small intestine and head over to the pancreas and talk about the nice things that it is doing for our digestive functions. And then we'll be ready to head back over to the small intestine and take a closer look at what's going on there during digestion.